Our final graduate student speaker today is Silvano Assanga. Silvano is currently a PhD student in plant breeding at Texas A&M. He obtained his bachelor's in crop science and master's in plant breeding and genetics from the University of Nairobi. Previously, he was a research associate at the International Maize and Wheat Improvement Center. While at Summit, he contributed to poverty eradication through development of climate change resilient maize hybrids for farmers in developing countries. He also held a position of a research officer in Kenya's National Research Program, where he helped develop nutritionally enhanced maize. Recently, he was awarded Monsanto's Beetle Borlaug International Scholars Fellowship to pursue his PhD in plant breeding. And please help me in welcoming Silvano to the stage.
Thank you, Laura. Howdy, everyone. I will talk about wheat, specifically wheat streak mosaic virus. We are doing genetic mapping on that, and later on I'll cover yields and yields components in wheat using the same population. So for those ones who are not familiar with wheat streak mosaic virus, it's one of the viruses in the genus Trichomovirus. It's mainly vectored by wheat calmite, which is indicated at the bottom picture. Although some studies have indicated that it is possible to transmit the disease through seed. The picture on the left side of at the bottom is a typically infected cell showing the virus particles, and this virus can move from one cell to the next through plasmodesmata, hijacking the cellular mechanisms of the cell to make copies of itself, further weakening the plant and causing the disease. Based on polymorphism of the coat protein, right now we have 131 accession types of the wheat streak mosaic virus that have been documented, and the information is available at the database of plant virus given on the slides. However, if you look in the United States, there's minimal diversity among the wheat streak mosaic virus. A study that was conducted by Assanga some time back indicated that it's easier to handle it in the United States. So in terms of geographic occurrence, it's a problem in North America, South as well as Central America, and some parts of Europe where wheat is grown. It mainly affects wheat, barley, oat, rye, but it can also cause problem in sorghum and maize. However, I would like to point out that in sorghum, it turns out that sorghum is not a good host of wheat calmite, and therefore it's not a big issue for them. Similarly, for maize, at the moment, it's not a big problem. However, it appears that if we have a double infection of wheat streak mosaic virus and maize chlorotic mottle virus, then the problem in maize is much more pronounced than even in wheat, where it causes a problem called maize lethal necrosis or corn lethal necrosis. And this is one of the major problems CIMMYT is trying to solve in sub-Saharan Africa. So in wheat, however, wheat streak mosaic virus per se has economic implication, and a study that was conducted in Texas High Plains, they were able to show that it can cause reduction in yield to a value as high as 76%. And in biomass, the value was as high as 6%. And all this occurs due to reduction in water use efficiency because the virus affects the root development as well as the shoot development. It is very difficult to phenotype for wheat streak mosaic virus because of the dynamics of the mites. Mites are moved by wind, so you cannot control where the wind will go. And therefore, it's very key to have some markers related to this trait so that we can help the breeding program to select genotypes that have genetic merit under wheat streak mosaic virus. And for this, our objective was to map some QTLs that are linked to the gene for resistant wheat streak mosaic virus. And the approach we took is to genotype a recombinant inbred line population using genome-wide markers. We used a SNP chip of 90,000 where we genotyped 217 recombinant inbred lines but we filtered out all those markers that are low quality and those that are monomorphic, not differentiating among our genotypes, and we ended up with about 5,995 high-quality SNPs that were used for downstream analysis. We did linkage mapping using join map, and QTL analysis we conducted using map-QTL. For phenotypic screening of wheat-streak mosaic virus, the 217 rails plus 3 checks and 3 susceptible checks and 3 resistant checks were screened in the growth chamber, and this was conducted twice in Kansas haze. And even though I said the virus is vectored by wheat calmite, when we did infection, we used a mechanical approach. Because our interest is in the virus, we couldn't use wheat calmite to transmit the virus because it's difficult then to separate resistance to wheat calmite versus resistance to the virus. So we opted to use a spray gun to transmit the virus to the recombinant inbred lines, and we took scores at 21 days and 28 days post-infection. So our work builds on the previous work at AgriLife Research, and the first genetic map for wheat-streak mosaic virus was published in Crop Science in 2011. And the nearest flanking markers, as you can see on your left side, I believe, it was 30 centimorgan and 45 centimorgan away from the gene. Clearly, this is not amenable for marker-assisted selection because you would expect then recombination to occur at that distance, which is too large. Later on, they followed up with a study to do a consensus map where they tried to narrow down the distance to about 5 centimorgan, which is still a little bit bigger given that there's no correspondence between genetic distance and physical distance. So in the current study, using 90,000 SNP markers, we try to zoom into the region where they mapped the gene. That's on chromosome 3B, the short arm. And within 2 centimorgan, from 3.0 to 4.9, we were able to identify 13 markers, giving us the degrees of freedom to identify SNPs that are tightly linked to the resistance to the gene. So we got an R-square that was high, about 91.1%, although this value could be higher given that we used a small population, so we could be having an effect, a BVC effect of overestimating the value. So these markers then were converted to another platform. Because we have few markers now, we wanted to test them. Do they work in a different genetic background? So what we did is to convert the markers to a uniplex platform, and we used Kompetitive Allele Specific Polymorphic platform to do that, which requires basically 3 components: the template DNA, the assay mix containing the primers, and the reaction mix that contains the fluorescent label. Now, this is a PCR-based process, so after cycling process, the plate is usually transferred to real-time PCR reader. Then you can be able to decode the information using the software and be able to tell whether the SNP can differentiate between the genotype. Now, I'll show you some of the results. The 2 SNPs on the genetic map are the one we have successfully converted so far from the Infinium platform to a KASP platform, and they were tested in a different background, and the picture in the middle shows how it separated the genotypes into 3 distinct groups. The middle group is heterozygotes, and indeed you would expect this if you're using recombinant inbred lines. It's difficult to eliminate the heterozygote or to attain complete homozygosis if you are using single-seed descent method to obtain rails. So it corresponds well with what we expected, and these are potential markers that can be deployed in a breeding program to select genotypes that have genetic advantage under wheat-streak mosaic virus. The picture on the right is just a confirmation from the Infinium platform where the same markers were genotyped. So there was similarity in either case. And in the middle picture, we had to make sure that we are not amplifying any noise. So everything was tested with a non-template control, and in our case, we used water in that case. So moving on, our second objective was to look at yields and yield components under drought and non-drought condition, because this is correlated with wheat calmite as well. The wheat calmites, they occur in warmer and drier areas. That's when you find the population is very high. So in this experiment, we phenotyped the recombinant inbred lines in 8 environments, where the environment here is a combination of location by year. And 3 of the environment were under dryland condition, and the rest were under a irrigated condition. They were carried out in Texas, Kansas, and Idaho, as well as one environment in Colorado. The trait we focused on were, obviously, yield is very important, heading date, as well as yield components of wheat, which are very key traits in wheat, such as spikes per meter square, mean single head weight, test weight, and kernel weight. We did analysis of variance, and the means we got were used to calculate best linear and bias predictors to try to shrink the means towards the true value so that can be applied for QTL analysis. And we used the same genetic maps we obtained from this 90,000 SNP chip to do QTL analysis. Statistically, all the environments we used, except one that is not represented here, was significant at 0.05, and entry mean heritability ranged from 0.3 to 0.8. Now, using the information from the phenotyping and genotyping, we did the QTL analysis, and I would like to draw your attention that the QTLs in these slides are color-coded by trait. So the first QTL are for grain yield, and those are 2 different environments and in different years, but the QTLs co-localized in the same region on chromosome 2b. Other QTLs that we found co-localizing with these QTLs were heading date, 3 QTLs from 3 different environments mapped in the same region of chromosome 2b, as well as QTLs for test weight and 1,000 kernel weight, as well as 1 QTL for plant height. The effect that was associated with QTLs ranged from about 7% for grain yield to 29% for heading date, as indicated by the blue bar. So, however, for yield alone, it ranged from 7% to 18%, given by the first 2 red bars in the slide. The additive effect ranged from, it was negative to positive. The negative indicates where the QTL is coming from. That, in this case, is a male parent, and the positive is from the female parent. The female parent we used is Colorado line, and the male parent was Texas A&M line. And indeed, term 111, as Jan indicated, is drought tolerant, and these results really agrees with that, because the effect is negative for the QTL for grain yield under drought. And that means that it's coming from term 111, which we know we have sufficient data that it's really drought tolerant. For kernel weight and test weight, however, the last 4 bars, we got mixed results where some QTLs were coming, the effect were coming from Colorado line, and some effects were coming from Texas A&M line. Moving on, we also mapped other QTLs for grain yield on other chromosomes, confirming how complex grain yield is. And these were localized on chromosome 3b, overlapping in a region of about 5 centimorgan. And in total, we had 4 QTLs for grain yield and 1 QTL for mean single head weight. However, I would like to point out that from the additive effect, it indicates all the 4 QTLs were coming from the female parent, which is the Colorado line. And the phenotypic variance explained by this QTL was less than 10% in all the cases. So it turned out that even though yield component is important for yield in wheat, they are totally located on a different chromosomes. And we mapped it on a chromosome 1A. A number of QTLs were on chromosome 1A, including mean single head weight, as well as test weight. And the effect ranged from 7 to about 11%. And most of the QTL were originating from term 111, except one QTL for spikes per meter squared. To summarize, we have developed a saturated map for wheat-streak mosaic virus, and the markers linked to the gene with confirmation, thorough confirmation. We think it can be deployed for selections of genotypes under wheat-streak mosaic virus. And we have QTLs, putative QTL for yields and yield components on chromosome 1A, 2B, and 3B. And markers linked to these QTLs will be converted to KASP platform and used to test genotypes that we have derived between spring wheat from Kenya and winter wheat from US. I would like to thank a number of people, our financial support from Texas Wheat Producers Board, as well as Monsanto Beecher Bollag Scholarship. We got some funding from the Bollag Leadership Enhancement Program that is administered at UC Davis, as well as the Bollag Institute from Texas A&M and the Critical CAP. We also thank the USDA for their support in genotyping using the 90K, as well as thank the term wheat genetic groups in Amarillo and the breeding and genetic group in College Station, as well as we appreciate the effort of undergraduate students as well as the graduate students that helped us in all this work. We also thank postdoc Travis and Soheb. They were very helpful. Thank you, and I'll take any questions.
Are there any questions for Silvano? Or if anyone on the webinar has any questions, please submit those now. There's a question over here.
Are the genes that you're looking at for wheat-streak mosaic heat sensitive?
Yes, so far we have two major genes for wheat-streak mosaic viral resistance. One was named WSM1. That's a translocation derivative that has been deployed in a number of experimental lines. And one commercial variety known as maize has been released that possesses that gene. It is sensitive to heat if it's exposed constantly to high temperatures. Now that one is not commonly used because the translocation from wheatgrass to wheat, it moved with other undesirable genes specifically affecting quality of wheat. Later on, they came up with WSM2 that was discovered in one of the female parent we used, and it still is sensitive to heat if exposed constantly to high temperatures of 24 to 30 degrees Celsius. So yes, they are sensitive. I understand, based on the literature I've reviewed, there is WSM3 that has not been widely tested and I think is still sensitive.
Okay, let's thank Silvano one more time.